Okay, here is the syllabus, and I have decided to not lecture at all on chapter 10 or chapter 11 or chapter 12. We're just not going to get to those. And I just don't want to push it. We'd probably have time to get some of the stuff from chapter 10 in today, but I just don't want to push it. So I'm not going to do anything from chapter 10 today. Um, so there will be nothing on the test next week that covers any of like the difficult experiments or any of like the deep ideas in chapter 10. I might have some of like the kind of simple terminology stuff from chapter 10 since you have read it. You've done those the Blackboard quizzes from chapter 10. I might have some like very simple kind of terminology questions from chapter 10. If that is the case, I'll make it very clear on the study guide what those questions would be and where to even find the answers. Or, like what I'm asking about, like kind of help you find in the textbook what I'm talking about exactly. That would only be from chapter 10. So the main chapters that will be covered on this final test would be 6, 7, 8, and 9. And there is plenty to talk about there, to ask you about there. So regarding that test, I will post a study guide for the test this afternoon. It will be up this afternoon, the study guide for test 2. Are there any questions about that, about test 2? It looks like we got something. Let's take a look here. Will the end of chapter 5 be on it? Yeah, that's a good question. I'll have to look and see. Yeah, it will be open all week. So you'll have like 5 days that you can take it. So if you want to take it Friday of next week, you have lots of time to fit it into your schedule. And yeah, it'll be up from Monday until Friday. At the end of the day, it'll be up all week next week. It'll be up on Blackboard. Will the end of chapter 5 be on it? Yes, I'll have to see exactly where we ended. That's a good question. I think we did end chapter 5 for test 1 somewhere in the middle. So there might be a couple things towards the end of chapter 5 that are on the test. I'll have to look. If I ask anything for chapter 5, it'll certainly be on the study guide, and it will not be lots and lots of stuff. I might have a couple things from chapter 5. There's a lot of overlap between chapter 5 and 6 anyway. But yeah, so if I do that, it will definitely be on the study guide. And there might be a couple things, 1 or 2 small things from the end of chapter 5 that could be on there. I will definitely make it clear. Any other questions or comments? All right. I am definitely happy to answer further questions. If you think of them later, just send me an email. Okay, so that's it for the test. I have extended the deadline, and the place where you can turn in your master and emissary writing assignment is now on Blackboard. The final due date is Sunday at 11:59 p.m. So I think that's Sunday the 9th at 11:59 p.m. is when that paper is due. I've definitely continued to be talking to people. Some people I'm meeting more than once, and I think it's helpful to talk things over with me. And I'm still happy to do that. So yeah, if you want to meet to talk about your writing assignment, that is a good idea too. So really, those are the last things we had. A in-class discussion quiz from chapter 8, and that'll be the last one of those. I'm also deciding not to assign the chapter 9 discussion quiz that I did have prepared to do, but I'm not going to assign that. So kind of the last 3 assignments of this semester will basically be that chapter 8 quiz, test 2, and the master and his emissary writing assignment, and that is it. So let's finish up chapter 9 today, which definitely has some interesting, difficult, kind of somewhat difficult concepts, and some difficult experiments as well, 1 or 2 of which we are going to be talking about today. So I have shown you this picture of the brain. You don't need to know all this anatomy for the test. It's not a bad idea to know that the hippocampus is located in the temporal lobe. The medial or middle temporal lobe is where the hippocampus is. The hippocampus is this blue thing right here. That's the hippocampus. That's the structure we've been talking about a lot. The amygdala is a structure we would have learned about a little bit more if I had decided to talk about chapter 10. I mean, you read about the amygdala some in chapter 10 anyway. It is notable that these two structures are right next to each other because they're probably interacting for things. We've already talked about the hippocampus is important for the encoding of long-term declarative memories. For instance, HM, that patient, after HM's hippocampus was removed, he was no longer able to form new long-term declarative memories, though he was still able to form new long-term procedural memories, apparently. For instance, I mentioned last time that he would not be able to do things like if you met him and then a day later and you introduce yourself when you saw him, you said, my name is blah, blah, blah. The next day you see him again and you ask him, have you ever met me before? He'll say no. If you ask, what's my name? He'll say, I don't remember your name, indicating that he doesn't have that long-term declarative memory. He can't form the memory and keep it, overnight until the next day. But that mirror drawing task, which is more of a measure of procedural memory, looking at something in a mirror, trying to draw its reflection, which is kind of a difficult procedural task. He did that over and over and over for several days, and he got better at it. If you ask him each day, have you done this before? He's going to say, no, I've never done this before. So he wouldn't kind of explicitly, consciously remember. His declarative memory wouldn't remember that he had practiced that task, the mirror drawing task, many times, but his muscle seemed to remember. So it shows this dissociation that even though his hippocampus was removed and he couldn't learn new long-term declarative memories, he still could learn new long-term procedural or non-declarative memories. So this pretty closely focused suggests that the purpose of the hippocampus is the encoding of long-term declarative or explicit memories. Oftentimes they're memories that can be verbalized. But there are a couple exceptions. For instance, we talked about animals. They have hippocampi as well. They don't verbalize anything. But we did talk about, I think I should actually show you that real quick. Where's that? Yeah, right here, the Morris water maze. We talked about the animals whose hippocampus was removed. If you drop them in the same location, they can get to this platform using their procedural memory. You drop them every time you drop them right here. This little rat could, if you drop the rat in this exact orientation, this rat could be like, okay, I just need to swim to the left for, a foot or two. I mean, they're not going to verbally say that to himself, but his muscles could know, I get here, I just need to swim to the left and then I'll be at the platform. That's if you drop this rat in this exact same location every time. It can use its procedural memory, even if its hippocampus is removed, to find the platform. But if you drop the rat in different locations each time, the rat can't just use its procedural memory. The rat just can't, if you drop it over here, the rat can't just swim to the left because then it would end up over here. It wouldn't be by the platform. If you drop the rat every time in different locations, the rat has to use its knowledge of these landmarks. For instance, the rat knows, oh, that blue square, the platform, which I like because it makes me safe, makes me feel safe, is located by the blue square. The rat needs to use the long-term declarative memory, its experiences in the past of knowing where that platform is relative to these landmarks. Presumably it would use long-term memory to remember that. If the hippocampus is removed, then the rat is not going to be able to encode that information into long-term memory. And so a rat with the hippocampus removed, when they're in the water maze, you drop them in different locations, they're going to have a lot of trouble finding the platform because they're not going to be able to use these landmarks to find the platform because they don't have the long-term memory of the hippocampus. They're not able to encode that type of memory into the hippocampus. So it's kind of like a long-term declarative memory for a rat. Kind of the caveat, kind of the interesting thing though, is that rats they can't verbalize, they can't declare anything, but nevertheless, the hippocampus is important for their memories, as indicated in the study. Their procedural memory was intact, though their declarative memory, as you might want to call it, because that's what it's called in humans, their declarative memory, when their hippocampus was removed, was definitely impaired. They could not use the landmarks to help them find the platform. So I talked about that last time, and there's kind of where those are the major bullet points, what I just said. Here's now a new study. It's also about the hippocampus. This is an imaging study, though. I'm pretty sure this one comes from your textbook. And it's a neuroimaging study, and it's a complex study for several reasons. One of the things that makes this, there's several independent variables, there's several factors, including one of the things that's also is complicated about the studies. There's kind of like two phases of the experiment. There's like a first phase where participants, they come into the laboratory and they perform a task, and they're also getting their brains measured there. And then later on, there's like a test phase where the researchers are measuring what did the participants learn during that first phase. So I guess the two phases, like the scan phase, that's like the learning phase, and then the post scan. This is the phase where the researchers are kind of assessing what did the participants learn. And as I said, this is an imaging study. So I guess I'll need to tell you a little bit about what the task was, what it was that the researchers were trying to teach the participants, so to speak. So what were the participants trying to learn during that first phase, the so-called scan phase? So I'll remind you, during this first phase, this is when the participants were being taught things. They're trying to learn something. Their brains were being recorded with an fMRI, I believe. And so it's important to note that the fMRI, the brain measurements, were trying to record, they're trying to measure encoding. So encoding is, the brain mechanisms which are involved when you're trying to acquire knowledge. You're trying to encode memories. And they were measuring that, what brain regions are involved when these people are trying to encode the memories. And kind of more specifically, they were asking what brain regions are involved when people encode memories that last all the way until the second session, that a day or a week later, whenever they come back into the laboratory. Those people will, some of the things that they were taught that first day, they'll remember. Some of the things they're taught that first day, they won't remember a week later. What does the brain look like when you're learning for those things that later on you will remember, compared to what does your brain look like when you're encoding, when you're learning stuff for those things that you're later going to forget? Is there differences, in other words, between the way your brain responds when it's encoding for things that you're later going to remember compared to things that you are later going to forget? And so the things that actual things that participants were taught on that first, the scan phase, the learning phase, to remember, had to do with these pictures right here. And the book does describe these in a little more detail. There's probably some stuff in the notes too, if I recall. I'll kind of give you a basic sketch of what happened in this experiment. So participants during the learning phase, scan phase, is like the first part of the experiment, would see a whole bunch of words. Just two of them are shown here. And basically the participants were kind of just looking at the words somewhat passively. They had to answer a question about the words. The question wasn't real. It was kind of just like a cover, like the researchers didn't, like, it was a very simple question. The question was something like, size and animacy judgments, not really important experimentally. Like if they would answer things like, is this an animal? Or is this a big thing, or is it a small thing? I believe that's what the size and animacy judgments were. These are kind of like a cover story. These weren't really that crucial to the experiment, but they wanted to make sure that the participants during this learning phase were actually looking at the words, that they weren't just ignoring things. So that's the main reason they had the participants answer these questions, to make sure the participants were actually looking at the words, actually reading the words. In order to do that, you have to ask the participants some question that they have to answer about these things. But do notice that the words are different colors. For instance, the word nickel here is green, and the word deer here is red. Ultimately, what the researchers are interested in is on that second session, can you remember the words that you saw during that first session? Do you remember, for instance, that you saw the word nickel? Do you remember that you saw the word deer? If I showed you the word book, that's a word you didn't see, maybe. Would you be able to say, I didn't see book, but I did see deer, I did see nickel? So that's kind of the first thing, remembering what words you did or did not see on that first day during the learning phase, the scan phase. That's kind of the first type of information they wanted you to, they were interested in if you could remember. The second thing they were interested for you to remember was the color of that word when you saw it. And that, by the way, is why I'm calling this contextual encoding. Kind of the context of this word. You're seeing the word nickel and it's written in green It's like, and it's a green, the word is green, the letters are green, the square that it's in is green, the word deer, the red letters, and it's like a red rectangle here. I kind of think of this as like the first time you meet a person, you remember the color of the clothes that they're wearing. It's kind of like the context in which you meet someone. You remember, a situation in which first time you met someone and you're remembering what they're wearing. That's what's meant by the contextual encoding here. I saw this word nickel and I remember the clothes that it's wearing, so to speak. That is the color that the letters of that word were written in when I saw it in the first session. And that's the second thing. That's the so-called source memory judgment, or the source judgment, that is performed during the post-scan phase. That's like the test phase. So, and here are like the two things during the post-scan phase that the participants were asked. They were shown a word in the post-scan phase, like the test phase, and they were asked, how familiar is this word? That is, did you see this word during the learning phase, during the scan phase? Have you seen this word before? And they were asked to answer on a scale of 1 to 6, where 6 definitely old means, yeah, I definitely saw that word in the first phase during the learning phase. And if it's like any other like a 1, that means no, I definitely did not see it during the learning phase. And they're asked also to report the color of that word. If they said, oh yeah, I saw the word nickel during the first phase. And the researchers would also say, okay, when we showed you the word nickel during the first phase, do you remember what color it was? And then the participants would, guess a color, green or red or something like that. Of course, in this case, the correct answer would be green. And that's reporting the correct color, once again, is the source memory judgment. It's kind of like analogous to when I met so-and-so, they were wearing a green shirt, or when I met this other person, they were wearing a red shirt. Are you able to kind of remember the context, that is the color that the word of the letters were presented when you originally learned it during that scan phase? During the scan phase, that first phase, the learning phase, that's when the participants' brains were being recorded. And what they wanted to see is, what is the brain for those words that on the second session, you are able to remember that, yes, this is a word that I saw previously. So it is familiar. You correctly say it's a familiar word that you saw previously. And you are able to remember the color of that word. That's like completely correct. Those two things, if you get both of those information, that means you really remember the thing that you saw that first day. You remember that you saw it, and you remember the color of the thing when you originally saw it. They wanted to see, what does the brain look like during encoding for those words that later on you're able to, what they called recollect. In order to recollect a word on that second session, that test session, you had to get both of these things correct. You had to say, yes, the word's familiar, I saw it before, and you had to get the color correct. What does the brain look like when you're encoding for a word that you later on will be able to recollect, that you are able to remember? Yes, I saw it, and I remember the color when I originally saw that word, compared to those words. What does the brain look like during encoding for those words that later on during the test phase you don't remember that you saw, and or you don't remember the correct color? So basically those words that you remember everything about them, what does the brain look like encoding for those words that you'll later remember everything about them, compared to those words during encoding that later on you're going to forget something about what you originally learned during that scan phase, that learning phase. And kind of the punchline is right here, those words that were later recollected, they showed more activation during the hippocampus during encoding compared to those words that the participants later did not correctly recollect. So maybe I should say this a different way. During encoding, if in this study, there were some trials where the hippocampus activity was big, some trials where the hippocampus activity was small. For whatever reason, there's fluctuations in the hippocampal activity during encoding, that first phase of the experiment. On those trials where the hippocampus activity was strong, a lot of hippocampus activity, the words that were presented during those trials, those were the words that were most likely to be correctly recollected later on. The participants, a lot of hippocampus activity during the scan phase, that encoding phase, the learning phase. Those words were later to be, yes, this is familiar, get it correct. And then, yes, I know the color of that word, and it was green, or it was red. And then if there was less hippocampus activity during encoding, you are more likely to forget that word. You are less likely to, say, yes, I've seen that word before, and or remember the color of that word. So this supports this idea that I've provided several pieces of evidence now that the hippocampus is clearly involved with the encoding, the formation of long-term memories. And this could be considered a long-term declarative memory quite clearly in this study, because you can verbalize the facts. The way you would verbalize a fact would be, yes, I've seen the word nickel before in this laboratory. And when I saw it previously, the letters of the word nickel were green. Similarly, yes, I've seen the word deer before in this laboratory. And when I saw the word deer previously, the letters that were composing the word were red. Declarative memories. When the hippocampus was highly activated during those learning phase, those were the words that later on you were most likely to remember, which suggests that the hippocampus is pretty clearly seems to be involved with the encoding of long-term declarative memories. Yeah, that's really all I'm going to say about this one. I get. Oh, there is these are just the brain activations. There's a lot going on here, but these are the activations from the study. These are the kind of the two parts of the hippocampus. I don't require you to know like there's like the posterior. I don't require you to know the microanatomy of the hippocampus in this class. So these are both basically for our purposes part of the hippocampus. These are the regions that when they were highly activated during that first phase, the scan phase, the learning phase, when these regions were highly activated, whatever words appeared during that part of the experiment, those are the words that later on participants were most likely to recollect. That is, remember, yes, this is a word I've seen before, and remember the color. If these were, for those trials where these words were decreased in activation, those were the words that people were later on most likely to forget, to not successfully recollect. I hear a microphone, which is fine if you're about to ask a question, but if you're not about to ask a question, please make sure your microphones are muted. Are there any questions or comments before I move on from this one? This will 100% be a question on the test. So if you do have a question about this, go ahead and ask now. All right, so the last thing we're going to talk about in this class is a neat theory, consolidation theory. And consolidation theory basically says that we know the hippocampus is important for encoding. But an interesting thing about the hippocampus is it's also involved with retrieval of memories. However, the involvement of the hippocampus with retrieval changes over time. So let's, like a simple example, let's say I formed a memory yesterday. I met a new person and that person said their name and they said their favorite, their favorite group, their favorite musician or something like that. That was yesterday. And then today I'm thinking about it. What's the name of that person I met yesterday? Their name was blah, blah, blah. What was their favorite band? And I'm trying to retrieve that information. Their favorite band was so-and-so. I'm using my brain to retrieve that information that I learned yesterday. And it's pretty recent information. I'm trying to retrieve a memory. It's a long-term memory now because it's 24 hours ago. And 24 hours is definitely in the time window that would be considered long-term memory. I'm trying to retrieve that memory from yesterday. And if you were to measure my brain activity, you would most likely see that the process of retrieving that memory involves a lot of activation of my hippocampus. My trying to retrieve that information of who I met yesterday and who their favorite band was from just yesterday would involve hippocampus. My hippocampus is highly activated in trying to remember something that I learned yesterday, a declarative memory that I learned just yesterday. When I'm trying to retrieve it a day later, hippocampus is highly activated. But this consolidation theory idea says that once I've learned something, the more time that passes, the less likely my hippocampus is going to be involved with retrieval. And that's what it says here. Involvement of the hippocampus in retrieval declines with time. And there is some evidence that actually supports this. I'm not going to go into detail about, well, there's actually, I am going to show you one study which supports this. I believe it's an animal study that we're going to talk about. But there's also some studies with humans that kind of that supports this idea right here. You learn something if it's encoded into a long-term memory, in those short time periods after it's become a long-term memory, when you're retrieving that thing, the hippocampus is really crucial. It's important. It's activated while you're trying to retrieve something. And so you can kind of think the hippocampus is kind of part, or it represents the memory that you formed just a day, maybe 2 days ago. The hippocampus is important for kind of reinstating, to remembering, to retrieving that thing that you just learned. But over time, if it's like, a week, or maybe even as far as like 4 weeks, much longer than just a day or two, a week, 4 weeks, so like 7 days or 28 days, the further away you get from that initial encoding of the name of the person and who their favorite band was, it turns out that the hippocampus becomes less important in retrieval. And then it's sometimes described as like the memory is kind of initially stored in the hippocampus for a small amount of days after it's initially encoded. But then it's kind of gradually the memory, you can kind of think of the memory is moving into those outer cortical regions, and it moves from the hippocampus to these outer cortical regions. There actually is fMRI studies in humans to kind of show this process a little bit of consolidation. That's what the consolidation is. You learn something, and for a few days, there is not too much consolidation, at least the kind of consolidation which is discussed here. The memory kind of stays, and it utilizes the hippocampus to retrieve that memory. But over a longer period of time, instead of the hippocampus becoming activated when you're trying to retrieve that, it's 28 days later, I'm trying to remember, who was that person I met 28 days ago? What was their name? What was their favorite band? Who was their favorite musician? And then if you look at my brain, then 28 days later, the hippocampus is not going to be involved with the retrieval of that memory. It's going to be more of those outer cortical regions. This is the idea of consolidation. The hippocampus is important for retrieval of a long-term memory soon after that long-term memory has been encoded. But as you get further away from the initial encoding of that long-term memory, the brain regions which are involved with retrieval are less the hippocampus, not very much the hippocampus, and more those outer cortical regions. The region's kind of the top part of the brain and the outside shell of the brain. That's consolidation theory. And I do have a experiment that suggests this. It is I believe this is an animal. Yeah, this is an animal study using fear conditioning. And so this is a study where the animals, basically the thing that they have to learn in this study is that when they're dropped into a certain environment, so these are like rats, so usually their environment is like a cage, maybe it might be a glass cage. There's different environments that the rats can be dropped in. One of those environments, every time they're dropped into it during a learning session, a learning phase of the experiment, the rats receive a shock. It says it right there. That's the learning phase. The rats receive a shock. Obviously, no one likes shocks. So if it's always the same context, it's always the same environment, it's always the same cage that causes the shock. And so the rats are going to learn, I don't like this environment. I don't like being in this cage because every time I come here, bad things happen. I get a shock. And so that's what the animal is learning, presumably. And the way you know whether, or the way it's inferred, whether an animal has formed that memory is by measuring the animal's freezing behavior. The idea, and it says here, animals, if they enter into an environment that they remember, this environment has previously, it's not an environment I like. Previously, I've been injured here. Previously, I've been shocked here. It's common for animals to demonstrate freezing behavior. That is, oftentimes an animal will explore an environment to kind of look around and see what's going on. But if an animal knows that it's an environment that has caused them problems, where they've been shocked before, instead of exploring, because they don't feel safe to explore, if they remember this is a bad place, they just freeze. They'll be dropped into the cage and they'll just freeze. They won't move, which intuitively that makes sense. If you're scared of a place, you're not really feeling comfortable enough to explore and get to know the place better, or smell around like rats typically do. So you can assess, you can determine, yes, the rat appears to, the extent to which a rat freezes, the extent to which it doesn't move, you can assess, that suggests the extent to which the animal, the rat, remembers this is a bad place. If the rat goes into a cage that has been shocked before and it moves around a bunch, then you can infer, okay, the rat does not appear to remember that this is a place where we had shocked him before. The rat does not appear to remember that he or she had been shocked before. And so I can show you, here is the graph from an actual published study. It is a little fuzzy for reasons I don't know why, but this is actually taken from a study. And the dependent variable here is freezing behavior. So like if the bar is really tall, then that means that when you put the rat into the cage where they had been shocked before, the rat does just freeze. But if the bar is small, like it is here, that means that when you put the rat into the cage, that they're moving around a little bit. They are doing a tiny bit of exploring. The taller the bar in this study, the more the rat is just stationary. The shorter the bar, the more the rat is kind of moving around. And remember, basically a really tall bar indicates that the rat appears to remember, this is a cage that has shocked me before. This is an environment that I don't like. I'm going to freeze because I'm scared. I do not feel comfortable to explore things here. And so there's well, I guess this is a factorial design. There's kind of two factors in this study. And there's clearly, by the way, a two-way interaction. One of the factors is, does the rat receive an injection of saline, which is a control? It's like, an inert chemical. It doesn't have any influence on the rat at all. It's basically water, salt water, compared to this chemical right here, CNQX. I don't actually remember what that acronym stands for. I do understand, though, that the purpose of this chemical, what it is believed to do, is to basically turn off the hippocampus, make it so that the hippocampus does not do what it's supposed to do. It's not activated like that it normally would be activated. The CNQX condition turns off the hippocampus. And remember, when we're thinking about consolidation theory, the idea is that when an animal or person is retrieving a memory that was formed recently, they're using their hippocampus to do it. And so that's what the researchers are interested in. The animal learns something, and then right before they test the animal, right before they put them in to a cage to see if the animal freezes or not, they are going to give one of these two substances into the rat. They probably inject it and then put it directly into the brain. Either saline, which does nothing to the hippocampus, or CNQX, which will basically turn off the hippocampus. And they want to see what happens to the rat's behavior. And they measure this. They measure freezing behavior either one day after the rat had learned what it would learn. The thing it learned, don't forget, the thing that the rat had learned is certain environments cause a shock. And certain environments are fine. The environment which these graphs are relevant to are those environments that did shock the rat. Lots of the environments didn't, but the environment that we're looking at here are those environments that did shock the rat. One day after the learning had occurred, or 28 days after the learning occurred. So that's the second factor, the amount of time that passed from the initial learning. One day after learning, that is one day after the animals will learn that this environment shocks me, and that's, I don't like this. I don't like this environment. When the rats were injected with saline, they still showed a lot of freezing behavior. But when the rats were injected with CNQX, which kind of shuts off the hippocampus, the rats showed significantly less freezing behavior. So their hippocampi were turned off here, which when that happened, they were more likely to explore, which suggests that they weren't afraid. When their hippocampus was turned off one day after the original learning, they had forgotten that this environment that they were in is one that they don't like, that just yesterday, one day ago, this environment shocks me. I should be freezing, but my hippocampus has been turned off by this drug. And so I forget, or I'm not able to retrieve this information that this is an environment which I don't like. This is an environment which is, I'm scared of. I shouldn't feel comfortable to explore here, but because the hippocampus was deactivated, I feel more comfortable exploring, and so I do one day after the learning. So the rat appears to need the hippocampus to retrieve that information that it learned one day ago. 28 days later, though, they do the same thing. The rat is either given the saline injection or it's given the CNQX, the CNQX injection, and then it's put into the exact same environment. And 28 days later, after the initial learning, the rat shows freezing behavior, a good amount of freezing behavior for both of these conditions. And a really notable thing, in this condition, the CNQX condition, the rat's hippocampus is deactivated. Nevertheless, they're still showing a good amount of freezing behavior. If you kind of compare the solid red bar to the solid blue bar, they're pretty clearly showing more freezing behavior 28 days later than they did in this session over here. The rats don't show much freezing. They're doing a lot of exploring right here 1 day later when their hippocampus was deactivated. 28 days later, their hippocampus is deactivated and they're freezing. And so you kind of think, why is this? And it has to do with consolidation theory. 1 day after the learning, they need their hippocampus to remember that information. If the hippocampus is deactivated, they don't remember this cage causes a shock. I should be freezing. They can't retrieve that information 1 day later after the initial encoding. And so they're exploring a lot right here when their hippocampus is deactivated. But 28 days later, now those outer cortical regions are the ones which are important. The hippocampus isn't that important because of consolidation. The memory has kind of moved to the outer regions of the brain. The hippocampus is no longer important to retrieve that information. And the information that they're retrieving is this context, this environment, this cage is bad because it shocks me. The hippocampus is deactivated, but it doesn't matter because they're not using their hippocampus anymore to retrieve that information. They're now using those outer cortical regions. So this supports, once again, consolidation theory. The hippocampus is involved with retrieval when a memory is newly formed. That's like the day one. But the involvement of the hippocampus declines over time. And by 28 days later, the hippocampus is no longer important for retrieving that initial memory, which is why for day 28, the rats are showing a lot of freezing behavior, even when the hippocampus is deactivated. So I do like this. This is a network theory explanation, or I guess it's called just like a network explanation for consolidation theory. The crucial kind of way of viewing, like this, you don't need to know this graph at all. The two kind of crucial graphs in this are right here. And these show what they call network dynamics. Basically, these are different regions of the brain, very simplified regions of the brain. You can kind of think of as the little circles as like, neurons or, parts of the hippocampus right here, for instance. And they say cortical modules. That's what I mean when I'm saying those outer cortical regions. Those are the cortical modules. If you want, you could be like, think of this is the frontal lobe. This is the parietal lobe, occipital lobe, maybe. Same thing over here, frontal, parietal, occipital lobe. And then this is the hippocampus, which is part of, once again, the medial temporal lobe. And all of these brain regions are involved to some extent when people, rats, are retrieving memories. But the crucial thing, once again, is when is the hippocampus crucial for retrieving a memory? And that's what's kind of illustrated in these two figures. As I said previously, the hippocampus is important for retrieving a memory when that memory is fresh, when it's just recently formed. But then over time, that's what's represented here. Over time then, weeks later, like 28 days later, 4 weeks later, the hippocampus isn't as important. And then those outer cortical regions are sufficient to retrieve a memory. And so that's what's shown here. And I'll kind of show like what these lines mean. So these lines are kind of, you can think of these as like signals which are being sent between these different regions of the brain. When a memory is freshly formed, like 1 day after you formed it, and you're retrieving that memory, the hippocampus, as I've mentioned, is highly involved in retrieving that memory. For instance, when I met a person yesterday, I'm remembering the name of that person and who their favorite musician was. I'm trying to remember that. I need my hippocampus to do that. And I kind of think of this as like the hippocampus is kind of like a conductor. In order to retrieve that information that I acquired yesterday, I need my hippocampus to kind of organize the activity of all of these different cortical modules. And so there's signals in the hippocampus which are being sent to all of these cortical modules. The cortical modules are sending those signals back to the hippocampus, and then the hippocampus is sending signals to other cortical modules, because there's kind of this interplay. The hippocampus is like the hub. It's the thing which is getting most of the activation here, but it's sending signals to these cortical modules. The cortical modules are sending signals back to the hippocampus. And then somehow, through the interplay that's going on between the hippocampus and these cortical modules, that is the process of retrieving the memory. But then consolidation happens, and consolidation, according to consolidation theory, is basically this idea that the hippocampus becomes less important over time. And the thing that becomes important for retrieving the long-term memory is those outer cortical modules, those outer cortical regions, become sufficient to retrieve the memory even without the hippocampus doing the conducting. The hippocampus over time becomes less important, and those outer cortical regions can do it by themselves. Early on, the outer cortical modules can only, or outer cortical regions, they can only retrieve the memory if they're interacting with the hippocampus. But eventually, the idea of consolidation is that connections form. You can see these red lines that are now existing between the cortical modules. If you look over here, there are no red lines that are directly connecting these cortical modules. In order for the modules to be connected to each other, they have to first send the signal to the hippocampus, and then the hippocampus sends the signals to the other cortical modules. But over here, over time, the idea is that connections form among these things by themselves. It takes time, but eventually these cortical modules become directly connected to each other so that you don't need the hippocampus to retrieve a given memory. And that is consolidation theory. And that's really all I have to say about that. Here are the questions I would have asked you. You might want to look at these because I'm not going to require you to turn these in. But I've mentioned before that the questions that I ask on quizzes, these are the, in-class discussion quizzes, in-class discussion questions. These are the concepts in the chapter that I consider most important. These are the concepts or the ideas that are most likely to appear on the final test. So I'm not going to require you to do these questions, but you might be interested in looking at these questions because these are the topics which are that I consider most important for the test. But yeah, I added here, I'm not assigning these to students. You do not have to turn these in, but there they are. And that's all I have for this question. I'm happy to answer any questions that you guys might have about any of this stuff. Otherwise, that is it. I will be posting the final study guide for test 2 tonight. So I'm going to stop recording now.